Dr. Jeremy Chien from UC Davis. How are you doing today?
Very good.
Great. So my name is Sarah Fisher. I'm a community engagement manager with the Cancer Genomics Cloud. You, of course, know me because we, this semester, well, you mostly, but with my assistants, put together a course for your students using the Cancer Genomics Cloud as a teaching platform. And so I want to hear how it went. I'm very excited to find out what all these final projects were like. And I want to know a little bit more about what your experience was like. So to start with, can you just explain a little bit about what this course was?
Yes. So the class is called First Year Seminar, and it is given to first year students. And in our university, we define these students as the student that come to the campus for the first time. So some of them are freshmen, and we also accept transfer students from community colleges and other universities. So those are also considered as first year students. So the class consists of freshmen as well as a few sophomore and a few junior. So these are the classes that are given to students, and they are designed to introduce these first year students to new topics that they might be interested in, so that they can learn about these different topics and find out which is the area that they want to go into. So these are usually short courses, like 1 or 2 credit hours, and designed for us to introduce topics that we are interested in, that they might be interested in learning more about. So it's also not high stake class, so it's a fast big class also.
So the students kind of self-selected into your section because it was a topic that they were interested in?
That's correct. So the topic that I designed for this first year seminar is essentially cancer genomics and bioinformatics. So if they are interested in cancer genomics or learning about bioinformatics, this is the one that they will be, if they are interested in how to do it, because the course description contains all these, outline, and also the requirement is that there is nothing that they need to know. Essentially, introduction. So that's the reason why, they self-select the area that they are interested in. And also this is designed to give a more hands-on experience and also smaller classes. So we limit the class size to the maximum of 18, so that they have more, I guess, more interaction with the instructors in the classroom.
So then tell us a little bit about what you did in this course. How did you set this up?
So, before this course was given, I have to submit it to the university about the plan. And so the idea was just to give them the general concept about the cancer genomics, essentially, introducing them the central dogma, and then design the course to learn about each of those, particular a domain like DNA-seq, RNA-seq, and then ChIP-seq. So that was the major plan in terms of designing, how we want to provide the experience about learning about these bioinformatics studies.
At what point did you decide that you wanted to use the Cancer Genomics Cloud as a platform for the students?
Yeah, very quickly, actually. So before, I was using Cancer Genomics Cloud. So I knew about the resources that are available, but I didn't know that this could be used for teaching as well. My students was using it for his own master thesis. So what I was thinking about planning for students about this class, I look into the institutional resources, and it's a lot more complicated. And then I saw the video that you have produced with another group from, I think, from Michigan, if I'm mistaken, maybe. So I thought, okay, maybe this is a really good idea because we are familiar with the resources, and the students will have access to it. So that's the reason why I started contacting you at very short notice, and I'm very grateful that you are able to provide your expertise and also a few lectures on how to use the Cancer Genomics Cloud.
That actually does bring me to one of the things that I really liked about this particular class. The way you're describing how you pitched it to the university and what you actually ended up doing in the class. This falls into the rubric of a course-based research experience for undergraduates, right? You had first-year students, some of whom I presume did not even have their full general biology coursework done. You had data that you produced in your lab that the students were able to just start analyzing. So wasn't that experience for you to be able to give brand new students your data and have them run with it?
Yeah, so we do not know what the results will be, right? Because this is the first time we are also looking at it. So as we finish the analysis, actually, if we have more time, it would have been much better for the students to see some of these results and then start critically analyzing it. Because what we saw was that there may be potential mislabeling of some of the samples. And we can clearly see that in our, cluster analysis and also the correlation analysis. But unfortunately, we didn't have this is a quarter system and very short time. So the student didn't have a chance to critically look into to assess whether or not there are sample mismatches in pair samples. They could be doing, RNA-seq because we had RNA-seq. We could also do the mutation analysis and then see whether the mutations are mismatching between the pair samples, supposed to be pair samples. So these are the things that, we could have done. But I was quite excited that, we were using this data, the fresh data, and hopefully next round, we'll be able to extend a little bit farther and then let them do the analysis and critically analyze these data so that they learn a lot more out of it. So I think that I like the fact that, we are using the data that we are freshly generating, but also the beauty of this platform is that you have also, the publicly accessible data. So if you don't have your own data, you can teach the course using the publicly available bit data. So that's the benefit of this platform.
Yeah, absolutely. So tell me a little bit about what analyses you actually had the students run.
Yeah. So we started with RNA-seq data, and we have 12 pairs of tumor samples. These are tumors collected from a patient with ovarian cancer, and we have the primary samples. That means they are before treatment, and then they have recurrent samples, so that's after treatment. So we have these pair samples from 12 patients. And the goal is to analyze the differential gene expression between those two groups. And then because we have a very deep sequencing on these samples, we can also do fusion analysis. So the first the learning objective for them was to perform RNA-seq analysis and do the differential gene expression and fusion transcript analysis. And then second project was to analyze the whole exome sequencing data. So we have a pair of tumor samples. That's a teratoma samples with the adjacent placenta normal tissue. And what we want them to do is analyze the whole exome sequencing results and identify the somatic mutations. So we did the basic analysis, and then later on ask them to analyze, how the ratio of transition versus transversions, and also annotation, and finally we want them to also learn about how to do the single base signature, mutation signature analysis. So those are the I guess objectives in the DNA-seq. And finally, the last one would be the last one we did was ChIP-seq, and that one also we have our own p53 the chip sequencing data from cell lines. So they did the analysis as well. In that one, we have the mutant p53 and Y-type p53. So I want them to know, what's the effect of mutation on binding to these target genes, and they were able to, identify what are the effect of these mutations. So that's essentially is the three major projects that we did.
That's incredible. I just want to sort of like recap that you had first year students who took raw data through an RNA-seq and differential expression pipeline and looked for fusion transcripts. And then they analyzed whole exome sequencing data. And then they analyzed ChIP-seq data. That's incredible. So, how did the students respond to doing this?
Yeah,
What did they feel like? How did they get out of it?
So I was quite happy to see the feedback from students. One of the students actually said that, when the student was coming in, they have, limited knowledge about what bioinformatics is. But after learning, all the steps, all the things that they learned from the classroom, they realized that they're just at the tip of the iceberg, that this is an immersed, subject that they want to know more about. So I was quite excited that they have at least some idea, some introduction to bioinformatics and cancer genomics.
That's great. Honestly, like as an educator, like if you get them to the point where they're like, oh my gosh, there's so much here that I don't know, that's when you know they're really learning.
So one of the students actually, approached me and asked whether or not there would be additional opportunity to learn more about it. So luckily, at our university, we have these individualized research studies for the undergraduate students. So they can register for that course, and then we can give them one-on-one instruction on the research studies. So this student is going to be doing the next step, which will be a single-cell RNA-seq analysis. And for that, we have data that is already published from another group, and we'll be analyzing that. And that is coming from 10x Genomics. And we also have our own single-cell sequencing data coming from Perth Bioscience. So this student is going to be doing that as another, I guess, individualized research studies.
That's awesome. That like they also like are going to go on to do more work with this stuff just because they were able to start to see this big world in front of them. That's awesome.
Yes. Yeah, I'm happy that we had this opportunity. I'm really glad that CGC has provided this platform for them to learn. Otherwise, I was thinking about canceling this course because it was just too difficult to teach. Give them the hands-on experience at using local institutional resources.
What aspects of the Cancer Genomics Cloud platform in particular did you find made it well suited for teaching?
So it's accessibility, right? I mean, everyone can access it immediately. And also the data, you could use the publicly available data and create the course contact really quickly. So that's the another benefit. And then the third one is, as they are doing the analysis, we can go into if they share the project, we can go in and look at it and how they're doing. So we can monitor the progress. And also if they had problems, we can see where the problem is, and we can troubleshoot it quickly. So there are a lot of benefits in using this kind of platform. The only drawback that I see is, because we are providing them, this common workflow language, and a lot of tools are available, and for them it is just point and click, and so it was too easy for them, and so they didn't learn, steps that are necessary or important. So as a project, in the end, final project, I gave them a project for them to do it by themselves without CGC. For that one, what we are asking them to do is just get the data, add in
Mhm.
results, for example, from RNA-seq, and then redo that locally with your own, the RStudio, for example. So they don't have to do the initials alignment and then
Mhm.
just use the count data and redo the analysis so that they at
Mhm.
least understand some of the important concepts that are important for them to properly learn bioinformatics.
How do you think you might approach it differently with that in mind?
Yeah. So one of the feedback I received from the student is that they actually like the Cancer Genomics Cloud platform. They like it so much that that the one of the feedback was that we should cut down the lecture and do more of those bioinformatics hands-on analysis because they find it really useful. I think so. Um, Going forward, what I'm thinking about doing is asking them to do more of those analysis, and then when we get the results, and then I want them to analyze the results at the point that we can talk about important bioinformatic concepts
Mhm.
so that they can incorporate the data analysis with the concept.
You have a master's student that you're working with in this class. And so what did you think your master's student actually got out of this?
A lot because he now know how to create packages and tools on the CGC. So that would be helpful because the next step, if I want to expand this course, will be for a little bit more advanced course where they will be not just using the tools, but also creating these common language,
Mhm.
workflows,
Mhm.
so that they can understand all the steps that are involved in creating the data analysis.
Mhm.
So it would be good to have that kind of TA involved and creating these packages
Mhm.
so that they can be involved in teaching next level of courses.
Okay, so that sort of begins to answer one of the other questions I had slotted to ask you. You are planning to try and expand to additional types of courses in the future?
Yes, definitely. I find this platform to be really useful, not just for research, but as for teaching as well. So definitely, I would be thinking about the next level of the instruction, where the students will be involved in not just learning about the tools, but also creating new tools as well.
The last thing I'm going to ask you is, what advice would you give to other like faculty members who are interested in using the CGC for teaching?
Yeah, just I would say, just do it. It is quite, it is, it may be quite daunting just to think about it, but when you start, you know, planning about it and then start interacting with the, people like you, they will learn that it is not as challenging or difficult as you think.
Mhm.
Because there are a lot of helps you can get from the CGC personnel. So I'm really grateful that I got the chance to work with you guys in creating this course. So if you are thinking about it, just do it.
Nice. That's
It's
great.
a good opportunity for students to learn about it. And they got a lot out of it. So I think it would be really good for the students uh, if this kind of courses are available everywhere.